here. mRNA is degraded by the action of small RNAs, and the protein is not produced. This is the basic mechanism. We have also an additional mechanism of transcriptional gene silencing, but I don't go in details with that. This is enough for today on the theory. So what is the or from where came the idea to use RNA as a fungicide? So we learn from nature. This here is a scheme which might be a little bit complicated, but I just want to show you. This is the fungus, this is the plant, and there is an exchange of RNA between plants and fungi. That was actually discovered earlier in mammalian cells that interact with parasites via RNA, but now we know it from 2013 on by Hailin Jin's group, showing that RNA is really exchanged between these organisms. And by exchanging the RNA molecules, they modulate defense reaction in the plant or virulence in the fungus. So this happens in nature. And the next step is just that we used, we followed the idea that we can use RNA to fight against viruses, insects, and nematodes, fungi, and oomycetes. And I say RNA, and there are different types of RNAs that could be used here. This is double-stranded RNA, but also small RNAs, circular RNAs, or even mRNAs. And as Nina Mitter already showed you, it is very, very nicely working against viruses. It is also working against insects, but there's a question mark behind fighting with RNA against fungi. I give you simply one example from our lab where it worked excellent on insects. Here, this is an aphid. Cytobion is a grain aphid, and we just feeded the aphid with double-stranded RNA, fluorescence double-stranded RNA in the artificial diet. And as you see, after 1 day, the fluorescence RNA is in the gut of the insects and then is distributed in the insect. So I don't go too much in detail with insects, but the efficiency of this RNA treatment, this double-stranded RNA silences genes in the insect, of course, is very different depending on the strategy you apply the double-stranded RNA. If you apply it via GMO plants, you have an efficiency of 80% on the survival. And if you do it with artificial diet, where they just feed from it, it is also very effective. But if you spray it, as you see here, if you spray naked RNA, it is not very effective, indicating we need a better strategy to apply the double-stranded RNA in the fields. So what is the issue here? I don't go in details, but we worked a lot on the uptake mechanism of RNA also in insects, and there are 3 possibilities I shortly show you here. There are receptors for RNA in the gut membrane here that transfer RNA from the gut lumen to the cells. Alternatively, there are RNA binding proteins that transport the RNA across the membrane. Do you hear it?
There is some disturbance.
Some disturbance is there, Dr. Kalyan. Can you check where? Kindly mute
everybody to mute and
only speakers. Let everybody be muted.
Narendra Singh has to mute.
Who has to? Everyone has to mute.
Dr. Narendra.
Okay. So I'm continuing. The third possibility for the transfer of RNA via the, across the membrane is here the Clathrin-mediated vesicle transport. So this is clear from insects, but as you probably, some of you know already, for instance, the RNA receptors are not present in mammals or even in fungi. So there must be another transport. I omit that because it's too long. So I focus now on fungus and the problem of the uptake of double-stranded RNA in fungi. We made a systematic approach by checking a lot of fungi whether they can uptake double-stranded RNA. This is a prerequisite for using this as a plant protection strategy. And I will focus today only on Verticillium longisporum here and on Piriformospora indica. I do that, but this is the Indian fungus here. Yeah, many of you know, of course, this is an endophyte fungus. It is beneficial, and I was interested in seeing whether the beneficial fungi are also affected by double-stranded RNA, which might be an issue if you develop plant protection measures. So starting with Verticillium longisporum and feeding RNA. So it is causing Verticillium wild on rapeseed, for instance. I don't go in details. What you see here is when we fed here if 420 base pair long double-stranded RNA, fluorescent labeled, you see the uptake in the hyphae here. This is very clear, so indicating the long double-stranded RNA is taken up. This is the second step then that you have to demonstrate, and that is not only uptake, it's also gene silencing by those specific double-stranded RNAs. And when we generated double-stranded RNAs against these genes, you see here, I don't go in details, different target genes in the fungus, and compare expression of the gene after treatment with in controls and double-stranded RNA against the target gene. There is a downregulation, meaning silencing. So it works. The 420 works. So we were interested to see whether there is a bias in the length of double-stranded RNA. Are some molecules more efficient than others, or is there a requirement for length as we found in, or is published in insects? But the answer you see here is no. There is no bias as far as we see, at least in Verticillium, where we fed 21, 420, and 1,451 base pair long double-stranded RNA, and you see the fungus took it up, and there was also gene silencing. So this is also the case with other fungi, but I wanted to stress the fact, as I said, checking these beneficial fungus. And here I want to stress again, this nice guy here, Ajit Varma, I don't know whether probably is not here in the audience, but he discovered the fungus, and this fungus is very, very interesting for science. And I'm working for 20 years on this Indian fungus, actually. So this fungus colonizes roots. As you see, I go very fast. We made field tests in Bali where we saw the fungus enhances yield, really yield, not only biomass, but yield. These are treated, these are untreated. And you see also effects on roots. So my topic today is whether this fungus takes up double-stranded RNA, and as you see here, time-wise here, incubation of the fungus with the spores of the fungus with 21 base pair long RNAs, the fungus takes up the double-stranded RNA, as you see very clearly indicating that beneficial fungi also do it, and this is important for considering this as a plant protection strategy. We also tested different lengths of double-stranded RNA again against piri, Formospora, and you see all these RNAs are taken up independent on the lengths. We have here after 2 days very strong uptake in hyphae, indicating it's very, very sensitive to double-stranded RNA. And even unexpectedly, if we tested very long RNA here, 1775, still you see a accumulation of this RNA into in the fungi. So I come to the end by just discussing if we use a double-stranded RNA and if we sprayed, we use the term exogenous double-stranded RNA. It comes from the outside. And the question is where it ends up in the plant if you spray plants. Not only spray, if you apply to plants. And if you here see an experiment with Arabidopsis, where we fed Arabidopsis via the petioles here with double-stranded RNA, and then we checked where is the RNA afterwards. And then for this experiment, we used 21 nucleotides that were labeled with Cy3. This is red. And we used Arabidopsis reporter line that visualizes the phloem. Yeah, the phloem is a SAC2 promoter in front of YFP. It's a reporter for phloem. As you see here, this is the phloem and there is fluorescence. And if we feed here the double-stranded RNA via the base, you see an accumulation of the RNA, but this is xylem. It is not phloem, but the xylem. If you see here the merge, you clearly see it's only in the xylem, the red color here, and it is not connected to the phloem, indicating that the RNA is not taken up in the symplast. This is naked RNA. Yeah, therefore we discussed yesterday with Nina Mitter that if you use formulations like BioClay, for instance, that might end up in the phloem. But if you use naked RNA, it is only in the xylem. The same is true if we apply it just by spraying or pipetting on leaves. Again, Arabidopsis that were drop treated with double-stranded RNA. Again, the 21 CYP3. And then you can see again in the reporter plant the phloem and the RNA in the xylem, and the merge indicates or suggests or proves that there is only accumulation in the xylem. So I'm coming to the end, of course, or already. For pests, the exogenous double-stranded RNA is a promising and very efficient strategy against selected pests, not all pests. I can't go in details with that here, but there are many, many things known already. Exogenous double-stranded RNA treatments in fungi, yeah, need to be improved. It's an innovation, but probably we need a lot of research into formulations. As I say here, and so far we see only a apoplastic transport from cell to cell or via the xylomatic route of double-stranded RNAs when sprayed onto leaves. Last picture here, sorry. I just want to give my thanks to all these people that are involved here. I don't go with the names, it's too long. We were supported by German industry here and by the German Research Council and the Ministry of Science in Germany. Thank you very much.
Can you unshare, please? Professor Kogel, can you unshare screen?
Yeah, yeah.
Thank you. Thank you.
Sorry, yeah.
Dr. Agarwal, should we leave the questions till the end, or we can ask?
We can ask here itself one or two questions,
Okay.
because they will be
Okay.
linked with the
Please.
presenter. Yes.
People can ask questions now.
Yeah. Can I ask a question, Professor Kogel?
Please, go ahead.
Yeah, Professor Kogel, you did give a good summary of this dsRNA in continuation to Nina Mitter's presentation. And obviously, I also happen to be an alumnus of University of Gießen. I worked with Professor Schlosser. When it comes to the uptake of this dsRNA, in case of fungi, what kind of mechanism it is operating? Do you have any idea?
Yeah, the mechanism is quite clear. It's RNA interference, and the machinery of RNA interference, like dicers and argonaut proteins, are required.
Is there any kind of endocytosis involved in this? Endocytosis?
Yeah.
Especially in fungi.
Yeah, but it's not clear. For instance, there is one publication showing now that the Clathrin-mediated uptake is required for the activity of the RNA. Yeah, so Clathrin-mediated uptake is one aspect.
Okay, thank you, Professor.
Any more questions, please? Anyway, thank you, Professor Kogel. It's nice to see you after 20 years, and I hope we will catch up once the COVID settles down in person. And I was really impressed with to hear the alternative technology to control plant pathogens. That's great.
Thank you, Professor Kogel. It was very nice, and maybe in future we will take some leads for fungal disease management through double-stranded RNAs.
It was a pleasure for me. Thank you very much.
Thank you for accepting the invitation and
just to
enlightening. Yes.
Just to continue, can we take something like electroporation as a delivery mechanism?
Dr. Desai, can we leave it till later, because otherwise we will not be finishing tonight.
Yeah, please go ahead.
It's my night. It's your afternoon. So I think let's communicate with Professor Kogel through other means. And communication, these are the points which we gave. This is the point of interaction, and let's continue with that. Anyway, Professor Agarwal, sorry, you're on phone. Anyway, it's my pleasure to introduce Professor Fernando. And by looking at this CV, brief CV, I can't say that it's very difficult to meet these kind of things and what he has done. And Professor Fernando, luckily, I visited your university once.
I see.
I visited the Winnipeg Rust Center, Cereal Research Center.
Oh, yeah.
And, but because of my previous 3 years postdoc on wheat quality, it was a great wheat quality lab in the University of Manitoba, Bill Busher.
Oh. When was that? Do you remember the year?
That must be 30 years ago. 40.
Okay.
Now, I visited there about 25 years ago, around maybe, but that was earlier. Busher was not there at the time.
Yeah.
But I think your impressive record of so many 165 publications, so many book chapters, and as dean and professor of plant pathology, and so many awards, CPS of outstanding research. Look, if I start reading, I will not leave time to present.
No, that's completely fine, Dr. Bariana. No,
I think it's you are one of the icons which our next generation should be sort of seeking guidance from. And so please go ahead and present your thoughts.
Okay. Thank you very much, Dr. Bariana, for that introduction. Thank you, Dr. Kogel, for that wonderful presentation. I had to get on line at 4 a.m. and I was worried that I may not be still up to give my presentation. But after listening,
to get up by that time, isn't it?
Yeah, it's so listening to Dr. Kogel was really good, so that I was able to get up correctly and have a cup of coffee and be ready. So it's again wonderful to be with the Indian Phytopathological Society's annual conference. I attended in person in 2016, and that was again a wonderful opportunity to meet many of my friends that I've made over the years at different conferences with Indian scientists. And I already see a few faces, Dr. Desai and others that I have come to know through my work and travel. So with that, I'm going to share my screen and get to my presentation. Okay, so today I'm going to be presenting, let me see, okay, there you go. So my presentation today, what I have named it as a new playbook in host pathogen interactions and resistance. So a new method that has been introduced through the research that has been going on for about 6 years in my lab. And I'm extremely proud of my graduate students, particularly 2 students who did most of the work that led to the R gene labeling, R gene rotations, and a postdoc who developed the Cas markers that I'm going to be talking to you today. So these are new tools in the toolbox that we have now for blackleg management. Let me tell you the story. Now, to get started with the story, I have to remember some of the people that have done phenomenal work in understanding how plant pathology should be brought into saving plants, saving crops, and providing food for the world. And people, all of you know Dr. Norman Borlaug, who worked in that particular region during the Green Revolution, Dr. Martin van Ginkel, Dr. Gurudev Ghosh, another Indian scientist who has done phenomenal work. I worked for a very short time, for a year, at the International Rice Research Institute, and I was very fortunate to meet with him and discuss things as a very young scientist. And then Dr. Glenn Anderson, a Canadian and from Winnipeg, where I am. Dr. Glenn Anderson, actually, the place that he worked actually was the Cereal Research Center. So he, along with Dr. Norman Borlaug, led the Green Revolution. So he was this, we call him the sidekick of Dr. Norman Borlaug. But all these people have done wonderful research that has led to not only an understanding of science, not only an understanding of what we do, but feed the world. Now, getting into the research, we are going to be looking at particularly vertical resistance in my presentation, but I just wanted to highlight that there's the horizontal resistance that we should always think about. That's something that is difficult to identify and put into a resistance background in a host. And because of that, it has been neglected. But with novel technologies, AI and GWAS, now it is becoming a much more preferable resistance to be incorporated into crops. Vertical resistance is by far the easiest to incorporate by breeding, and that's what I will be focusing on in my presentation today. So to get started with the plant resistance, I thought just wanted to also show why it is so important, especially in a country like Canada, United States, I'm pretty sure in India as well, there are large acreages owned by one farmer. And so, for example, if the farmer owns 2,000 acres, the applications of fungicides and other methods that might be used might take a lot of time, energy, and also need a lot of money. So plant disease resistance requires no action by the farmer during the growing season, is not disruptive to the environment, is generally compatible with other disease management techniques, and most often, but not always, is a very good method to singularly suppress plant pathogens. So my presentation is going to be involving around canola. And for those who may not particularly know where canola was initially developed, I just wanted to give credit to who we call the father of canola, Professor Baldur Stephenson, who was from my department, Department of Plant Science at the University of Manitoba. So he developed the first variety, the variety called Tower, and that was in the 70s. And around the same time, Dr. Keith Downey at Agriculture and Agri-Food Canada in Saskatoon also developed. So we give credit to both of them sometimes and call them fathers of canola. Today, the crop is about $30 billion each year. So it's a huge crop that is very much valued. And in 2002, our breeding team, myself and Dr. Peter McQuitty, were honored with a Canadian stamp for the research that we had done over the years to develop canola. Now, why is canola so important? You can easily see this from this slide, that over 90% of the canola grown in Canada is exported as oil, seed, and meal to markets around the world. So only about 10%, because the Canadian population is about 37 million, we do not consume too much. I mean, the primary source of edible oil and also meal is canola in Canada, but we don't need so much that because 22 million acres are grown each year. So most of it goes to other countries. So that tells us who dictates the crop. The crop is dictated by other countries saying you do not need, we do not need a certain disease and so on. So when it comes to diseases, we have to play the game on the trade side of things. So blackleg has become a trade issue. And this in particular has made the work to be done on blackleg and resistance and mitigating the disease an important factor. So I won't go into detail here, but I just wanted to show that every part of the plant can be infected. The least amount infected is the pods, but the leaves, the stems, and everything can be infected. The name for blackleg, Leptosphaeria maculans infection, comes from what happens right on the stem here that girdles the plant, and there will be plants lodging. Now, looking at the disease has been increasing. If you look at the blackleg prevalence, it is about 73% in Manitoba in 2018. And when you look at the incidence, it's about 13%, but even 13% can be sometimes devastating to an economy. Anything over 1% to 5% can be devastating. So this is a very important disease that we have to manage. Now, as I mentioned, it's a huge trade issue with China. We do send about the worth of $3 billion of seed to China for crushing, and not for planting, but it has become an issue with the Chinese government that we have blackleg, they don't. And because of that, the persons who travel are the Prime Minister of Canada, Mr. Justin Trudeau, and the Trade Minister would go to China to discuss this because it's a huge issue to deal with. So some of the reports that Mr. Justin Trudeau and the Trade Minister carry are from my lab, but it's at the diplomatic level that they discuss in mitigating the loss to Canada. And on the other hand, we also have the loss to the farmer. And this is shown with on the right-hand side with the amount of disease that might occur, the severity will form what is going to be at the field level. So market access is as a issue that we have to be addressing most of the time. So in resistance, we all know this quadratic check. The host genotypes R genes should be matching the A virulence genes of the pathogen. It's a lock and key mechanism of the receptor and effector. And in all other situations, there will be disease. So this is showing from the canola plant, the seedlings. Now, since 1970s, when blackleg first appeared in Canada, we have done extremely well with good genetics and breeding to keep the disease under control for about 30 years. But in 2002, we started to see an emergence of the disease. And this is a picture taken in 2011. You can see a variety that was commercially available as resistant was performing like a susceptible variety in the field. So this is where I wanted to bring the story of what we are doing and talk to you about how, so for example, if A virulence gene, AVRLM3, the matching RLM3 interact, what you get is a hypersensitive reaction. This is a classical plant pathology. And then RLM3, the same gene, the same seed can be now diseased when it is being attacked by the virulent strains of the AVRLM3 and end up with disease, with susceptibility. Now, in 2010, we understood that we didn't, even though we were doing screening and we had identified that there are certain varieties that are resistant, some are not, we realized that we didn't have any understanding about the resistance genes in the Canadian canola varieties. Now, some of the work that had been done in France they had been able to identify some of the R genes much earlier, because the disease of blackleg has been in Europe for a longer time, and it has been researched more than in Canada. We have had a very much cushy period of about 30 years with good genetics and not particularly having an issue. And so one of my PhD students did this research in identifying all the R genes in Canadian germplasm. So all the companies participated and we were able to identify the R genes. At the same time, another master student in my lab was able to identify the AVR genes, and you see that the A virulence genes are at about 3%. So 97% of the AVRLM3 are virulent. So you can just imagine what really happens in the field. Even though RLM3 is present in the crop, in the variety, it is not acting as RLM3 because the virulent isolates are going to have causing disease. So further to this problem, we have found that AVRLM3 in the presence of another gene in the same chromosome, AVRLM47, this particular gene will mask the activity of the AVRLM3's function and give disease. So in addition to AVRLM3 being virulent, even the presence of AVRLM3 sometimes is not working because of the presence of the AVRLM47 and giving this phenotype. So we call it aggressive mimicry. The presence of a functional allele of AVRLM47 hides the RLM3's mediated recognition of AVRLM3, which should have been like this. So this is a new trick we call, we don't know how long this trick has been, maybe for 1,000 years, we don't know, but it is one of the things that we in science, we also call epistatic suppression of AVR recognition. So when you look at this, so we were interested to really see how many of these are present as AVRLM3, but not showing the phenotype. And here's the PCR. So when you do genotyping, 2014, 15, and 16, the isolates of AVRLM3 showed this amount of high percentage of AVRLM3 presence in farmers' fields. But when we do the phenotyping with the same isolates, this is what you get. You hardly get the presence because the phenotype is masked by the AVRLM47. Because AVRLM47 is at a high percentage on these isolates, it is masking the effect of the pathogen. So some of the work that has been also done in Australia, we took cues from Australia. They have shown that if you grow the canola variety on the same infected stubble of the same variety, or in other words, the same virulent form, you get a lot of disease. But that same variety, Hyola 50, can be grown on a different stubble, which means it's a different virulence genes, and then you will not get the same infection. So taking this as a principle, we had done research to see whether that would become a true fact in Canada, and all the preliminary results showed that. So we went ahead and advised the canola industry that we should come up with a resistance group labeling. So because now we had identified the genes that are present in the Canadian canola cultivars, we were able to come up with labeling the genes. This labeling is mainly for the farmer so that they don't have to worry about the RLM gene when they go to the retail shop to buy. So what you find here is, once you know this, the farmer can make that decision as to which variety they should be growing. So any time that you have grown a variety with group A genes, that is RLM1, you should not grow that in that same field the next year. Why? Because you are having a lot of inoculum if there had been disease. So if in the when there has been a lot of disease, you should not be growing that. You should be able to grow all the other different groups, B, C, D, and sometimes when there are stacked genes like CD, BCD, and so on. Now, when A is again there, it may be a okay rotation because the B and D might be helping in reducing the disease, but also there might be certain amount of disease because A might be already going to be broken down because of the presence of the AVRLM1. So this is mainly being brought in because a lot of farmers, because of the high cash value that they get from canola, they tend to grow canola in tight rotations. We still ask them to grow other host crops in the rotation, but that sometimes doesn't happen. Now, this is a fine example again from Australia showing when you grow group D on group D stubble, this is what happens. But that same stubble, group D, can allow group E canola to grow with the group E GR genes, and you will get a fantastic crop. So this is again a very good example to show you that the disease can be managed if you know the R genes. Now, to make it further easy for the one of the questions that you might have in your mind is, so how does the farmer come to know what AVR genes or virulence is present in their field? To do that, one of my postdocs working, Dr. Kaveh Gambania, working in Dr. Hussein Bohan's lab, was able to develop Cas markers that would separate out and identify the virulence from avirulence. So these Cas markers were shared with seed testing laboratories, and with the seed testing laboratories testing this for a very nominal fee for the farmer, they would get a good understanding of what is out there in their field. So once they know that, they can go to the retailer and ask for a variety that did not have the virulence, the matching genes, and can get a variety that would be a good rotation variety for their field. So here now also on the other side, the companies started to label their varieties now that they knew the variety, the R genes. And here's an example from Monsanto. Now it is Bayer Crop Science, owned by a German company that shows the resistance is there, but the resistance is carried by 3 resistant genes. So this is a stacked gene variety, RLM1, 3, and S. So here's an experiment that we did in the field just to show what I had earlier shown in the Australian situation. Here, RLM3 on RLM3. The RLM3 is the most predominant in Canada. And you can see that the crop is completely diseased, premature maturing. The same field, if you have RLM3 stubble, but now you grow RLM4, you get a fantastic crop. So this is a clear indication that by understanding the genes, you can manipulate so that the pathogen is not in a position to cause disease. So here's another example from the same field, having 2 different varieties, one carrying only the RLM3, one carrying RLM3 and RLM4. So that's a stacked gene situation from the same field. And you can see that when you had RLM4 was actually protecting RLM3 from breaking down, and there was a significant clean crop in this field. This was during COVID. We were fortunate to go do some experiments in the fields, and this is from Coronation, Alberta, taken on September 23rd. So to end my presentation, I wanted to give you how resistance can be approached with this kind of knowledge that we can, once we have done research in the lab and greenhouse, recommendations can be done for the field. So this is from Dr. Rob Fraley, who said data by itself is useless unless you can turn it into recommendations to be used on the farm. So this is, I think, the most gratifying thing for the researcher. So we publish, but I think at the end of the day, what we have done, once we see that the grassroots, the bottom line is being affected, impacted in a positive way, that's when we really see the value of research. So with that, I would like to end my presentation. As I said, all this work was possible because of the fantastic collaborations we have, the funding we had, and also the students and postdocs who worked on it. Thank you very much.
Could you please unshare screen, Professor?
Yeah.
Great talk. And I was a bit disappointed in the beginning. You didn't say Australia, because black leg is a big issue here. And I'm in Australia, although I'm Indian born. And
so most of the pictures that most of the pictures I told you were shown from Australia. From Horsham.
And it's nice to actually understand the dilemma which causes, like, it's like humans. One person can protect the other. You can say AVR genes are also doing the same. And it's quite a nice, fascinating story. And I don't want to come between you and some of the people who might like to ask questions. Great talk. Really appreciate it. And I must say that I've noted down, and I took few shots as well, but I will not share with anybody, but just for my own sake. Right?
Okay.
Any questions, please?
Well, Chairman, sir, I have one question.
Yeah, please.
Yeah, Professor Fernando, okay, it's excellent to contain the disease with the gene environment.
Something is breaking up, my dear. You can put it on the chat. Are there questions on the chat now?
I think you can put in the chat, and then we can later discuss.
Yeah,
With the permission of
sure.
the chair.
It was an
We can
excellent.
move it. Yes.
Yeah.
Dr.
Yes, if you
Bariana.
permit me, Dr. Bariana. It was an excellent presentation by Dr. Fernando, and the molecular work leading to the practical aspects. He very well demonstrated that how this AVR gene in presence can even reverse the susceptibility. That was really wonderful. And
Exactly,
maybe
I
it's
think-
leads for many, it's a concept he has proven, and then it can be really followed in many other host and the genes where we have already seen that what genes are acting. What are the susceptible genes?
I
Yes.
should take that opportunity to say something, Dr. Agrawal.
Yes, yes, sure.
Thank you for bringing a very important word there, the molecular work.
Yes.
So a lot of people sometimes have a disconnect between molecular work. They would say, that is not going to have an impact on any of the research. That's not going to be solving problems in the farm. So everything that we did was done through molecular. Of course, there was phenotyping, but everything that was done through molecular work, Cas marker development, all the PCRs from cloning, everything. So you can see how it has an impact if you really need. Thank you very much for
That's
being
excellent. Both the presenters,
Sorry,
like previous present.
just one thing
Sure,
I want to add. Can I please? Professor Fernando, I think the approach which you have brought up pretty much like lab to land. If we do anything which can go to the farmers, that's what we need. You know, we can publish good papers, but we need to take, we have to prove the things and then take it to the farmers. And I work in the Australian Cereal Rust Control Program, and we are pretty much like that, we actually identify genes, but we are reliant on phenotypic and genotypic technologies. And I am happy to see all those colored things which our pathologists produce for the wheat varieties in Australia. It was great.
Can I ask a small question to Dilanta?
Hi, Dr. Desai.
Hi, Dilanta. How are you? It's a long time.
Very good. Yeah, very good to see you.
Yeah, one thing I just wanted to ask you, because in Australia also, drought is a major concern.
Yeah.
So what will happen to this kind of resistance when the crops face something like abiotic stress?
Oh, yeah, very good question. Now, first thing that I need to mention is the drought. We generally don't have the same type of drought as Australia does. So that's number one. When you take the three provinces, Saskatchewan, which grows the highest amount of canola, is the driest out of the three provinces. Both Alberta and Manitoba get substantial moisture during the growing season. And also, particularly because of the snow melting away, the initial period where blackleg is infecting has enough moisture. So, so that period is what is the most critical to control from a biotic stress standpoint. We generally do not have abiotic stress on canola. Even in Saskatchewan, we don't have that. So it's a good question, but at least if the global climate change has not happened, we don't have that. Yeah.
So I think that's a good discussion, Dr. Desai, which can take, because Canada is actually, moisture is always there in the beginning.
Yeah.
And the other thing,
that's why I specifically mentioned about Australia.
If you actually look at necrotrophs, are much difficult to manage than biotrophs.
Yeah.
And in necrotrophs, we will be learning more from the GWAS and other things, but we have to keep Army and Air Force together. I call horizontal as Army and vertical as Air Force. Horizontal as Air Force and the other one as Army.
Yeah.
So we have to put them together.
It's a very good work, Dilanta. Congratulations.
Oh, thank you. Thank you very much.
And with the time, because time is going very quickly.
Yeah.
And I have to introduce next speaker. Do I need to? Soash.
Thank you very much.
I have known
Youngsters, youngsters.
so long, you know. I always thought that I'm actually older than Swash, and he turned out to be proving me wrong. He's few months older than me. Anyway, that's all right. But he doesn't show gray hair. Swash has done a wonderful job, as Professor Kogel said. Dr. Nagarajan's legacy, he has taken that baton he's carrying for last. I wouldn't be talking about your awards. You have meant so many awards. NAS award. NAS is a bad name, and but it should be called NAS. NAAS. And although so many publications, Swash, you are a very enviable scientist in India, and you have been part of sequencing mission with, I know, the current DDG, Dr. Sorry, remind me the name, sometimes I forget. Dr. Sharma. And I think what you have done is basically what Professor Fernando said. You have actually always taken the wisdom from lab to the land. That's what your group is actually known for. You have told farmers what the pathogen is doing, right? It's a great work that you have done. You have led, and you're still leading until you retire, and probably be passing the baton to another younger generation. But I really am proud of you. And you have LR80 in the beginning, and that's what we work together on. And we are really proud that Australia-India collaboration has given us quite a bit of platform to achieve things together. And I'll leave the stage for you now.
Thank you very much, Harvans. It's a pleasure. I mean, you were so much generous in introducing me. And I think it was a wonderful collaboration during all these years, maybe more than 25. And it is always a pleasure working with you, because you have been so helpful and so cooperative that we could get this new gene LR80 also. So I would request Indian Phytopathology to present my slides, because I have already submitted along with sound.
Okay.
So present my slides, please.
Okay, Dr. Bharat, just do it.
So I think nobody is, unaware of wheat rust. If you see the losses caused, so they can be 100%, and these are pathogens worldwide. And I think wheat is main crop, so it becomes definitely of everybody's concern that we are able to manage this. So I'll be presenting here almost knowledge which was gained during last.
Share the screen, please, because it's not. I can't see the screen.
Yeah, they have to share. Actually, they must be in the.
Just wait, sir. I am sharing from here. Is it coming now?
Yes,
yes,
but you have to put it into the slide operation. Yeah, it's great. Thanks.
Voice is very feeble, Dr. Kalyan. You can increase the volume.
Okay, I can increase. Thanks. Can you hear me? It's all good. I could see it.
Yes.
I could see. Yeah, it's fine. I can see it.
I think it's not audible.
Not audible, yeah. Voice is not coming, Kalyan.
Check whether Actually, that icon is not coming. That's why I'm searching. That volume can be controlled.
Swash, do you have
No,
a talk? You can
the
unmute.
voice
can
is
just
coming,
say.
but very feeble.
Very feeble.
Yeah.
Dr. Kalyan, you can
Bhardwaj
go
can
with
speak.
the slide.
Yeah, now, sir, Dr. Bhardwaj, you can continue with the
So,
live presentation then.
like Harvan said that this is definitely a race going on between breeders and pathogens. So we are having resistant variety. It is becoming susceptible. So again, we develop resistant variety. So here I just want to say that how does evolve in India? So we trust they do not have any alternate host like Australia and many other countries. So evolution is more the help of you can say mutation. So mutation that is very high because we have got say a high production of spores in the case of rust. And if one mutant is there in one, say 10⁻⁻⁷, so many mutants can be there. So when it comes to management, so as my predecessor said, Dr. Harvans also said that we'll have to go for resistance breeding because that is definitely a hassle-free and you can say minimum cost measure for controlling wheat diseases. So here I can just say that when there is a single gene, so chances of its breaking, that is nearly 10⁻⁷ or 10⁻14. And here I have just said about the different types of resistances which we have: race-specific, race-non-specific, you can say seedling, adult plant, and then adult plant race-specific, it is hypersensitive, then it is non-hypersensitive, then we have got non-race-specific, what you call slow-rusting genes. So I can have next slide, please. So why single gene should be avoided? Because behavior of genotype, if you see when we evaluate even in seedling, you can see many of the genotypes, they show resistance, which is not slow resting. These have got number of pustules, very few, and then more latent period, more incubation period, and low terminal disease severity, and effectiveness of genes that varies with area, because a gene can be effective in Australia, but it may not be effective in India. And the slow rusting, that is again, I mean, a good tool. So when we calculate area under disease progress curve, then it is 101 to 200, I mean, the rating which we get. So these are the slow rusting varieties, and definitely we do not get statistically say significant losses when we have got slow rusting genes in our varieties. Next, please. So next slide, you can just please load. So this is the slide which I borrowed from Dr. Ravi Singh from similar to that, how, I mean, when number of genes that increases, how can you have nearly resistance when you've got 4 to 5 resistance genes? So that's the story. I mean, putting more number of minor genes and getting very less of disease and definitely no significant losses in yield. Next, please. So when you see plant breeding approaches, we have got different approaches, but for resistance breeding, that's backcross method that is the best. But you can have next slide. But you can see that nowhere in the world we have got targeted breeding for rust resistance except for few countries. But it is haphazard breeding mostly. So this is the way how do we select a resistant material. And here I have said that when you go for double that time in say desirable resistance that you can say population, so that's how you can select. So this is backcross breeding, and but in conventional breeding you require a number of generations, number of population, that is more quantitative traits cannot be introgressed, and recovery of recipient genome that is not ensured. And it poses difficulty for negative selection, that is genetic drag and other problems. But if you, next slide, please. Then if you go for that backcross breeding, you can see that we can minimize the number of population and then generations also, because here you have got a selection backed by marker, tightly linked markers. And but the best thing is that if you go for screening evaluation, that against different pathotypes in different locations, definitely you can have a better choice. Followed in one of the project where we have introgressed 2 genes. So that's how we proceed 2 genes into one variety, and DBT we have done. We made crosses, and ultimately we made double cross, and that's how we selected. So this is the genome recovery in the different cases, the red genome and the blue genome. That is very clear that how much recovery of the genome that was there from the agronomically superior parent. So next, please. Can I have next slide, please? So this is again benefits of that I call it a marker assist. I think it is being pushed further. So marker assisted breeding, because you are taking help of markers, so number of generations that is minimized, and definitely you are focusing of your choice of gene. LRAT we have done it in a collaboration with Harvans. So it is a new gene where it is effective against all the pathotypes of leaf rust in India and Australia, and it's the 6th gene in the history of 74 years of resistance gene designations, and we have designated 223 resistance genes. And this is the, how do you differentiate this gene from other genes? So this is resistant against all the pathotypes except for 16-1, which does not infect bread wheat, but it infects tetraploid wheat. So this is the beauty of this gene, that it imparts. So this is here you can say on one side, these are SSR markers, and on other side, we have got a tightly linked CASP markers. So if we go for integration of these gene into new varieties, definitely will have, say, a very good selection, because we have got a tightly linked markers. Next, please. So take home message is that for breeding for rust resistance, you must have more than 1 gene or 2 or 3 genes, and definitely assisted that backcross breeding, that's the best method, and you should go for searching new genes, and combination of seedling, adult plant, and hypersensitive, non-hypersensitive, then slow rusting genes. Another strategy which has come up recently, which even Harvans is also practicing, that with one major gene you combine one slow ruster, so that is going to be, an ideal combination. And definitely, as Harvans says, we'll have to live with the rust, because we have seen it even in case of that right translocation, also new pathotypes, they came up. So we'll have to manage them, but definitely we'll have to live with them. So that's all. Thank you. Thank you very much.
Excellent. Thank you, Swash. And I think, as always, this area, I can't ask questions because we have been discussing almost every day these kind of things. WhatsApp, thanks to WhatsApp, And but I just want to add one thing. Professor Fernando mentioned some big names, and there was one missing, and it's Dr. Rajaram, who passed away a couple of months ago, or maybe last month. And he was standing with Borlaug in the field when Borlaug's Nobel Peace Prize was announced, and he's alumni of alumnus of University of Sydney. Definitely. And he trained Martin Van Ginkel.
So alumnus of IRI also.
Dr. Professor Bariana, thank you so much for bringing that name, Dr. Rajaram. In fact, I met him at the International Congress of Plant Pathology. I had the pleasure of introducing him to the audience, and also he was very kind. He gave one of his books to me. He's so passionate about everything that he did, and I'm extremely sorry to hear that he passed away, because that is news to me.
Actually, he was still continuing breeding. He was continuing breeding while things were stopping due to COVID, and he got it, and he was recovering. And after that, some complications came in.
Oh, sorry to hear.
So it was pretty much about a month or 2 months.
Just 1 month back, just 1.5 months back. And in recent, I'll just share with you, in recent convocation, which we held, IRA held on 12th of February. He was conferred with honoris causa Doctor of Science degree.
Oh,
Yeah.
Right.
And he accepted it, and we also wanted to make him an adjunct professor, which he said because of little issues he won't be able to join. But we are so fortunate that he accepted our this request, and he received.
And his varieties are doing so well. Even
So
now,
well,
he's
yeah.
a private breeder. You know, I would say that I would give him some credit for the Green Revolution.
Yeah,
Yes,
2018,
of
I met him
course.
at Boston also, and he joined that meeting also.
Harbans,
ICPP, yeah.
Harbans, I want to share today's summit named Taluka Station in the name of Dr. Rajaram.
That's great.
Excellent.
Wonderful. Very
Yes,
good.
today.
So very, very nicely today we are remembering him.
Excellent.
Yes, thank you.
Excellent.
Thank you for bringing that name. It's very fond memories. 2018 in Boston to meet him.
Yeah.
He on his own came back to where I was seated, had a good chat with me, and gave that book. So I will never forget him. I'm so sorry to hear.
He was always nice, and so does Gurdev, Kush.
Yeah.
I mean, you won't imagine. I talked to Rajaram about 3 months ago about farmers' agitation, and at that time there was no sign that he will be sick. And Gurdev Kush also, I'm in touch with.
Yeah.
Anyway, we have to close the session, otherwise we will never close it. Thank you very much, all the speakers. And you have enlightened us with the wonderful thoughts. And I think Professor Kogel, double standard RNAs is our armory to actually, I think, keep up with good work. And I think we probably need more more of that work going to defeat our enemies. No, it takes time, but you are a persistent player. You will do it. You will achieve it. And Professor Fernando Blacklag, what you presented was really great. And I think as an Australian, and India, I don't know about blackleg. I can talk about wheat rust, but it was a great, really, data you have shown. And Swash, as always, I don't have to admire you. You are great. And I pass on to Professor Agarwal. And
yes, thank you, Dr.
I'll stay to the
yeah,
end.
you stay. Yeah, you also will be with me throughout. So wonderful. So we have finished the key speakers. Now we will invite the invited lecturers. Thank you all the 3 key lecturers, and very nice. So under the invited lectures category, we have Dr. Robin Gogoi, the first speaker.
Yes, I'm sharing
Okay.
from here.
Okay.
So he'll be presenting from here.
So he is presenting on unfurling the Medes blight resistance in maize germplasm of India. So he is handling all this e-conference very meticulously planned, and also he's a maize pathologist.
Only desktop, direct desktop.
Dr. Robin is doing wonderfully in the division. He's doing integrated disease management on maize. He will be sharing the slides.
Yes, ma'am. Just
okay.
Yeah.
Welcome, Dr. Madhuban Gopal.
Yeah, sure. I'm here.
I could see
I'm enjoying your chairmanship also, and I enjoy the lecture especially. And hats off to Fernando, Dr. Fernando. He's awake at 4 o'clock in Canada, Manitoba.
Yeah.
By the time Dr.
Yes.
Robin is putting his slides, I'm just wondering, you know, he's the lone person working in IRI on maize. He's traveling and he's working in IRI also. Let's see how much is the progress made as compared to Canada and Australia and other places, because he's the
So I
lead.
think it is now seen, the slides.
Slides are seen. Come to the slide mode, Dr.
Okay.
Mandal. Yeah. Yes. What happened?
Actually, it is not going back. I think so many.
One slide. Let him start from there only here.
Okay. So start.
Good afternoon. Am I audible?
Yes. Yes, Dr. Kulkarni.
I think I am visible. Also, my slides are visible.
Yes. Yes.
Honorable Chairman, Dr. Harbans, joining from Australia, and Dr. Rashmi Agrawal, and also renowned plant pathologist, joining from different parts of entire world, what I should say, also from our country. So today, I'm going to present what actually I am doing here. Part of my work. It is not the whole work. So under the title, unfurling the resistant mechanism or resistant character in the maize germplasm of India against maize leaf blight disease. So as we know that maize is known as cereal queen, and this is third important cereal crops. So it is very popularly used not only as food, feed also, then for biodiesel also it is popularly used. In terms of production and productivity, so India accounts for 5% of corn acres and contributes about 2% of global production. So this is the disease, maize leaf blight disease. You can just imagine these are severe cases of this symptom of maize leaf blight disease. So it is distributed almost all the maize growing parts of India, and under severe cases, since it is called blight, so it is completely burning. So this maize leaf blight disease is caused by the fungal pathogen bipolaris maize. It was also known as Helminthosphoria maize, and this sexual state is Cochleoblast heterostropus. So, but in the Western countries, this disease is known as Southern corn leaf blight disease. And under severe cases, from 30% to 70%, the yield loss have been recorded. So for this disease, actually, I have separated my presentation into 4 parts. First one is confirmation about the race of the pathogen bipolaris maize under Indian context. And second part, what is the actually resistant the plant characters for which some of the plants are showing resistance to this fungal pathogen, and some genotypes they are showing susceptibility. This is morphocultural characters of the pathogen. And one always debatable issue or doubt was there. See, bipolaris maize is having some races, and race O, C, T mainly. But under Indian scenario, so it was earlier known as race O, and over time, so this race profile may also be different. So for that purpose, to know, to ascertain the what actually the race prevailing in Indian conditions, so we collected 75 different isolates of bipolaris maize from different agroclimatic zones of India where maize is grown. And based on the morphocultural characters like conidial characters, cultural characters, colors, texture of the colony, so that we just made some of the dendrogram, and we found out of the 75 isolates, including Delhi isolates, Karnal isolates, Ludhiana isolates, where actually this disease is dominating, means northern India, this disease is more dominating. So 6 clusters we have got. And to go further from each cluster, so we have taken representative isolates. Total, we have selected 25 isolates for detailed study. And based on molecular characterization to know the ascertain the pathogen, whether it is bipolaris medis or not. So we performed ITS sequencing, GPDS sequencing, actin gene sequencing, and also beta-tubulin gene sequencing, and it was perfectly showing some distinct identification, and phylogenetic analysis was also done using all the 4 gene sequences. Here in this dendrogram, I have shown for 2 only, and on that basis, so you can see this accession number also we have collected from gene bank, and for each ITS, GPDS, actin, and beta-tubulin genes, and we submitted. And based on the ITS characterization, some of the important isolates we have deposited in our Indian type culture collection to obtain the accession number. So it is confirmed that this is nothing but the bipolaris medis. Now, next part regarding the identification of the specific race. So it was actually long time demand to know the racial profile of bipolaris medis under Indian condition. So here, with the help of our maize breeders, we got very good germplasm, which are the cytoplasmic male sterile lines. Here you can see the categories CMS-S, CMS-T, and CMS-C. These are our cytoplasmic male sterile lines, and this is CM119, highly susceptible to this bipolaris medis disease, this pathogen. So it is having normal cytoplasm. And here you can see the very, very higher score with respect to 1 to 9 scale of disease score. So very high score you will get in normal cytoplasm. That means in case of CM119. And on the other hand, in other cytoplasmic male sterile lines of 3 categories, is S, T, and C. So only few infection, even C was remaining almost free from of the disease. You can witness from these photographs. This is C, CMS-C, this is CMS-T, this is CMS-S, and you can see we are heavily infested. This is normal cytoplasm possessing this inbred. This is CM119. So based on the various this inoculation technique and using 25 selected this isolates of this bipolaris medis, so we could prove that this is only race O, because this race O is infecting only the normal cytoplasm but not infecting the CMS lines, none of the 3 CMS lines. So this is also we have confirmed last year only. Based on this genotypic characters, histopathological and molecular characterization also we performed. So here 2 groups ultimately we have got. Here this resistant germplasm in bed lines, and these are susceptible lines. Here CM119 you can see, and on the other hand, first 2 lines SC7 and SC24, these inbred lines were also developed here. This elder maize pathologist, and it was both were registered in NBPGR, and is highly resistant. So we carried forward our next research works using highly resistant inbred line of maize is SC7 and highly susceptible CM119. And if you see the plant characters, in case of resistant germplasm, so many these leaf hairs were found, which are known as trichome. On the other hand, in susceptible the CM119, this less number of these trichomes were counted. Similarly, with respect to the number of stomata in resistant one, SC7, less number of stomata count could be recorded. On the other hand, susceptible CM119, many stomata were recorded. And again, we performed this scanning electron microscopy and transmission electron microscopy. And this infection pattern of the pathogen, these are the mycelia of bipolarized medes, how they are actually penetrating through the stomata. Here, more proliferation is visible, proliferation of the bipolarized mycelia as compared to the resistant one. On the other texture, this wax deposition in the resistant one was found higher than the susceptible one. Similarly, mitochondrial these structures. So in case of resistant inbred lights, they were almost intact. On the other hand, this mitochondria were So with respect to the physiological parameters also, we found the higher—
Is everybody mute?
Yeah, higher photosynthesis synthetic rate in case of highly resistant SC7 as compared to the susceptible one. Similarly, stomatal conductance was less in case of highly resistant one, and transpiration was also less in case of highly resistant SC7 as compared to the susceptible CM119. And when we performed by utilizing 50 different assessor markers, so out of that, 2 markers we could get which showed highly polymorphism, these 2, UMC 1590 and Y1. And out of these 2 SSR markers, we have found that comparatively UMC 1590 is reliable, which can be utilized for this breeding purpose. In another one, so development of the population. So in this case, we have done this crossing between the highly susceptible CM119 and SC7 highly resistant one. And in the F1, selfing was done. And after F1, so F2 was developed, and next BC, 2 more populations we obtained. This is BC1P1 and BC1P2. Means BC1P1, we have done this back crossing with the susceptible parent, and P2 crossed with the resistant parent. And we got the population. And in that case, so out of these 6 populations, namely P1, P2, F1, F2, BC1, P1, and BC1, P2, they were evaluated for their performance against MLB disease under artificial epiphytic conditions in 3 different locations. One is Delhi, another in Karnal, and third one is Dholakwa of Himachal Pradesh. And in this slide, you can see this among the we have performed this testing the goodness of feed for the segregation of ratio of F2 generation, and then we found the chi-square values for 15:1 ratio. And among the epistatic interactions, and dominance was found positive and significant across the 3 locations. And further, dominance genes affect across the test locations that indicated the presence of duplicate genes. So here you can see that the segregation analysis and the prediction of the number of major loci revealed at least 2 major genes associated with MLB tolerance in subtropical maize of India. So after that, again, we performed some of the gene expression study by utilizing qPCR method. So first one is phenylalanine ammonialized activity. So we assay, and here in this case, if you see this red one is resistant inoculation, and the main one is resistant control, and this orange color is susceptible inoculated one, and yellow color is susceptible control. So in this case, phenylalanine pal activity was actually increasing, and also gene expression was recorded high, initial for up to 48 hours, and also it reached to the highest in 72 hours, and again with the advancement of time at 96 hours, it reached to the peak. So another one, pheophytinase gene, or it is also called as chlorophyllase gene, means this gene is involved in the degradation of the chlorophyll content in the folia parts. So the role of this gene also was studied. So in a simplified way, we can tell that this expression of this chlorophyllase gene was less in case of resistant this is SC7 as compared to the highly susceptible CM119. So another PR protein beta 1,3 glucanase also we actually tested for its expression, and you can see here this that's in case of highly resistant inbred SC7 at 48 hours it was increasing. Again, it is decreasing at 72 hours, and also in 96 hours, it was that expression was highest. So it is fluctuating, it is not stable. So we can conclude overall from this gene expression study that you see the genes defense and basic metabolism which interact during the symptom development of MLB disease in maize plants, and also indicated their association with the interaction stages of bipolaris maize infection, and also respond to the resistant genotypes, resistant as well as susceptible genotypes in the disease, course of disease development. So this in a 4 different way we studied about the resistant mechanism which are prevailing in case of the highly resistant inbred lines as compared to the highly susceptible inbred lines under Indian maize genotypes studies. So I must acknowledge here the division of plant pathology, and the head also Dean Dr. Rasmi Agarwal, she is very much present here. Her support is my strong support, and I must acknowledge IRI, and also this NAHEP cast. One of my students received that fellowship from NAHEP, and my working hands. Actually, what work I have presented now, this is 7 years work. So 4 students, Venkatesh, Manjunath, Jivan, and Shweta. So I must acknowledge all the 4 students for compiling my presentation and executing my ideas with the help of these 4 students' hands. And thank you for patience hearing.
Thank you, Dr. Robin. Very nice, very significant research highlights you presented. 2 significant works you highlighted. One is the presence of race O. Now we are very clear that in India, bipolaris maize race O is present. And second, you highlighted how what is the basic mechanism behind resistance in maize resistant lines in comparison to susceptible lines. So now it is open for discussion. Some quick questions can be taken. Dr. Harbans, you have to add anything or
yes, sir.
Say anything. Dr. Harbans Pariana. Yes.
Unmute, sir.
Unmute.
It was nice to see CMC coming, that cytoplasm coming very nicely, protecting your, in your experiments.
Yes.
So it still goes back to the old stories as well. You can say that cytoplasm can do it. And great work. Keep it up. And I remember your name almost every week because one of my collaborators on CRISPR-Cas is Neelam Gogai.
Okay, thank you.
Great.
I remember you every day.
Thank you, sir, for compliment.
Any other observation?
Madam, I have.
Sure. Yes.
Sir, this is Jeevan. Very good afternoon, sir. So you have characterized bipolaris isolate. You have collected around 50 isolates, sir. You have characterized based on different genes. So
my student, yeah.
So my question is whether you have characterized based on the single gene or it is a combined gene analysis, or any other single gene can able to differentiate those isolates. That is my, because
we have to take care. But this presentation based on seeing a single gene, but we have to go for multiple genes.
Yes.
Okay, if there are no more questions, thank you, Dr. Robin. And we will go to the next speaker. Next speaker, Dr. Shekhar Shetty. I request all the now speakers that kindly restrict your time, because already we have, it's already going to be 4:30 and we have to quickly finish our presentation. So next speaker is Shekhar Shetty. I think for the invited speakers, as organizers say, it is 12 minutes for the invited speakers. Am I right, Dr.? So kindly restrict. Shekhar Shetty. Yes.
Even we can limited the questions.
Question answer. One question.
That's exactly
nobody. Yeah.
With earlier 12 minutes. And I think one of the things is that Madam, sorry, I'm
Yes,
taking.
no problem. No,
One of
please.
the things is that if you are given 12 minutes, finish in 10, because you want to be the professionals, not to tell the whole story. Just tell the cream. Bring the cream out.
Yes.
And that's important. If you go to the international symposia, overseas, so that you should not be seen as you are going over time.
Yes.
Yes.
So I request everyone now, the invited speakers should take 10 minutes and 2 minutes for discussion.
Yes.
And all the oral presenters should take 8 minutes plus 2 minutes for the discussion part. So kindly bear with us and follow the timeline. So Dr. Shekhar Shetty will be speaking on innate immunity in plants and systemic acquired resistance for plant health, challenges and opportunities to ensure food security. So Professor
So
Shekhar
Dr.
Shetty,
Shekhar, are you sharing your slide or shall we share from here? Dr. Shekhar, Professor Shekhar Shetty.
Sir, Shekhar Shetty is there, no?
Professor Shekhar Shetty there.
Is he there? I could not see. Are you there, Dr. Shekhar? I have my doubts. No.
So, ma'am, without any delay, I think
yes, we can proceed to the next speaker.
Yes.
Dr. Mahendra Singh Saharan.
Yes, madam.
Yes, another my very good colleague and wheat pathologist, and extensively worked at different centers. And I will say his contributions to wheat program is wheat pathology program is monumental, and he was earlier PI of the All India Coordinated Plant Protection Program when he was at IIWBR. And in 2016, I think he came to IRI, Division of Plant Pathology. So everybody knows him.
Dr. Saran, you will be sharing from your side?
I am sharing.
Can I add one word?
Yes, sure, sure, Dr. Harbans.
Long
time colleague.
Mahendra is more like a younger brother.
Again, emotional.
And he was part of our Indo-Australian project for 10 years.
Yes.
And he was wonderful, and I think he made the program successful. And he was the key backbone about that. And, I congratulate you moving to the Teaching Institute, and keep up with good work.
Thank you, madam. Thank you, Dr. Harbans.
Yes, please.
And good afternoon to all.
So, and Dr. Saran will be speaking on head scab. Now it is an emerging disease in India. So a very significant work he is carrying on.
Don't go over time, please.
Yes.
So I am presenting on studies on occurrence, variability, and sources of resistance for head scab of wheat in India. I am audible.
Audible.
I am audible.
Yes, Dr. Saran, you share your screen. You are audible. You are very much audible.
Okay, I shared my screen.
Okay.
So
we cannot see Dr. Saharan.
Yours. Otherwise, I will share from here.
You share from your side.
Quickly do it. Yes. Already 1 minute we have lost.
Just.
So, has it come?
Yes, it is coming.
Okay.
So, I am presenting studies on occurrence, variability, and sources of resistance for head scab of wheat in India. Next, please. Next, please. You are hearing me?
Mandal, please run the
slide.
I can hear you, so, Saran.
Yes.
Yes, actually it is, just a minute.
You go.
Not responding, so that's why I'm trying it. So maybe maybe slide or maybe whatever is.
I think Dr. Sharon can share and do that because
yes,
it will be hectic.
So it has come.
Yeah, yes. So we all know wheat is important crop in India, and this is second most important after rice. And go to next, please. Go fast. There are 6 wheat growing zones in India in different zones due to climate. There are different biotic stresses problem. So different disease in different zones. And this is just I want to tell that more than 450 varieties have been released in the country since 1965. And this yield production of wheat has increased from 6.4 million ton in 1950 to more than 107 million ton last year, and this is likely to increase further. Next, please. Next, please. Next, please.
I'm trying.
What happened?
Sometimes
not going. Sometimes get stuck, huh?
Get stuck. That's what I'm.
So you can quickly go on passing to the next slides because it's all in his tips. Dr. Saran can speak even without the slides also. You got stuck, huh?
Got stuck, ma'am. Actually, I'm trying, but yeah.
I think the system has the problem. So better, I think Sharon can do it.
Yes, these are the different states of India, and you can see that headscape is present in all the zones now. Earlier, we were getting sample only from the whole valley or Wellington, and this is due to the global climate change that is going to be important, and more important is the mycotoxin production, DON, ADON, etc. So these are different terminology used for the disease in different countries, like in Canada, called FHB, in Europe, headscape, like that. Next, please. So during 2005, when we were on survey in Punjab, there was a variety PDW274. So that was severely infected at that time, and durums are more susceptible to headscape. We noticed that. Next, please. So these at different places in Himachal Pradesh, we collected many samples. From Wellington also we got. Next. So this year also from Wellington, from whole farm, from ARI Pune, Dr. Vikas was on tour there and he got these samples. In MP also we are getting now. So this is the status of headscape in India. From 1974, first time reported by Rai, then Burma and Singh from Wellington. And likewise, now it is present in Northeastern Plain Zone, in Southern Hill Zone, Peninsular Zone, in Northern Hill Zone, in Northwest Plain Zone. So it is coming now more. And during this year, there was more incidence of headscape. So sometimes we mistake with wheat blast. Wheat blast is not present so far in India. These are the wheat blast symptoms, but in headscape, there will be pinkish growth in the spikelets. Next, please. So from Lahore Spiti, we got at that time during 2005, these 3 species mainly, and these macroconidia which are present for the Fusarium graminearum. And next, please. So we did the RAPD, and on the basis of RAPD, we characterized the 32 isolates of Fusarium graminearum and other Fusarium species. And on that basis, we could characterize these isolates. Next, please. So we did also developed also 4 microstyles markers while I was working in Switzerland in Professor D. Pago lab. And these, on the basis of these microstyles, we could characterize these Fusarium graminearum and other Fusarium species isolates. Next, please. So this is also fluorescent labeled microstyles we developed, and this paper was published in Crop Protection. Next. So to know whether in Indian isolates there is a gene which is responsible for the production of mycotoxin, we use the tree 5 primer, and which is can tell us whether gene associated for tox5 and tree 5 is present or not. Next, please. So in our isolate, we through using HA tree and GJ tree, we could know that there are genes present in some isolates of Fusarium graminearum. These are the isolates from Wellington, where we can say that the gene is present there, and it is the matter of the environment whether toxin is produced or not. Likewise, for GZ3 also, there was amplification with Indian Wellington isolates. For pathogenic variability, we studied this on the basis of the spike infection. Next, please. Then this percent spikelet infection also, on the basis of this from Wellington or from Lahore Valley isolate or Gurdaspur isolate, we could find that there is variation in the disease level in different varieties. Next, please. Likewise, we also did the area under disease progress curve, and on that basis, there was variation in the disease progress. Next. Last year also, we could collect many samples from Wellington, and scientists from Wellington also helped Dr. Vikas and Dr. Nalathambi there. And these are the 29 isolates, and we characterized these on a set of host lines. And there was some isolates were more virulent, while some were less virulent. So this some isolate like FGW10 and FGW24, they were more virulent. And we designed some 23 SSR primers. And from that 21 SSR primer amplified these Fusarium graminearum isolates. And on that basis, through SSR, we could characterize genetically these 29 isolate which we isolated last year from Wellington. Next, please. Now we have developed the system evaluation procedure by injection or by the cotton web technique. Cotton web technique gives the better result, and this we are using. Next, please. So this is the assessment procedure in the poly house. We evaluate the material. We have evaluated more than 5,000 lines, and on that basis, the resistance is very less in Indian material. Next, please. So this is the disease developed even now in this season at IRI in the poly house. This is the level of the disease. Now recording is going on. And these are the some moderately resistant sources we have identified. And we also use Sumai3 and Frontana. And we have developed some lines which are now at IIWBR. We also developed some resistance genetic stocks. Two genetic stocks, one with bread wheat, one with durum wheat. And these have been registered with NBPGR. And there are some elite lines which can be used further. Now this one Switzerland line, Swiss line CH, this we also found resistant to our isolate. Next, please. Next. We also evaluated some Meritini lines, and we could find that some lines are resistant, which are developed by Dr. Vinod here at IRI. So these are some other sources which are present in other countries like Sumai3, Ling 7840, Puno. So this is an overall view of the resistant sources available other than India. Next, please. Next. This is just some sources which are available in other countries. Next. So I want to conclude that there are mainly 6 Fusarium species, and Fusarium graminearum is the dominant. And this genetic and pathogenic variation has been studied, as there are very few resistant sources to head scare in Indian material. So there is a need to evaluate more number of wheat germplasm. Thank you very much.
Thank you, Dr. Saharan, for excellent presentation. Dr. Harbans.
One thing, I think Professor Fernando is still on. He sent me an email, very encouraging. And he said he works on FHB and not on Rust. But I think Mahendra and Professor Fernando, I work on land races and I've got genotyped populations I'm happy to share. You know, if you guys can check the parents and something comes out, I'll be very happy to share.
I think
Thank
this
you.
is a great opportunity.
Great.
Thank you, Dr. Saran, for that presentation, and Dr. Harbans for that message. I think these are the opportunities that we get from conferences, and this is an opportunity. So, Dr. Saran, as Dr. Bariana mentioned, I work a lot on fusarium head blight as well. So I'll be more than happy to collaborate with you in any aspect that we can, and also with Dr. Bariana on those things that we can do together on wheat and wheat scab.
Thank you,
Thank
Dr.
you.
Fernando.
Excellent.
Thank you very much.
Very good.
Yeah.
Thank you, Dr. Harbans.
Anybody has any quick question, or otherwise we'll move? Thank you, Dr. Saharan. A wonderful
Thanks,
presentation.
Madam. Thanks, madam.
I will request Dr. Harbans, the next oral session. You can
Yes,
begin
please.
with the Philomene Juliana from CIMIT. Yeah, you know.
Lectures.
We probably have met few
Please,
times.
Mariana.
Yeah.
Yes, Philomena, we probably have met few times in conferences. You may remember me as the RAS person. And I know you work with Ravi and the whole team of Sridhar and the big man, the other guys. And I think your work is very much appreciated, and you have done some real good publications recently. And I think now we want to hear
Dr. Kalyan, do you find her there? Dr. Kalyan?
I'm also not finding her.
I wonder she has maybe some
Let's
technical
move, and
issues.
we can do it later. I was
Yeah.
introducing them for
Okay,
nothing.
yes. Next.
Yes.
You can proceed, Dr. Bariana. You can proceed with the next speaker.
Is she wrong?
I could not see her.
So,
you can do the introduction for the others. I'll do Vikas because
Okay, no problem. Dr. Neeraj Dwivedi. I would like to invite Dr. Neeraj Dwivedi from Energy and Resource Institute, New Delhi, and he'll be speaking on parental polymorphic survey and screening of F2 mapping population derived from a cross between Bhut Jolokia and IV, another genotype against anthracnose in Chile. So it's a very important problem, anthracnose in chilies. So we would like to hear Dr. Neeraj Dwivedi.
Dr. Neeraj Dwivedi.
Is there? Are you hearing Dr. Neeraj? Maybe he's thinking in between. He's connected, but Dr. Neeraj.
Dr. Neeraj, can you share your slide?
Madam, if we can move to the next. And
we will move to the next. He joins because he is muted.
Yes.
Dr. Nanjundan, he is also our reporter. Yes, Dr. Nanjundan, you please go ahead. He will be speaking on identification and characterization of Indian mustard accessions with novel resistance against multiple isolates of Albigo candida occurring at Wellington. Wonderful. It's another problem.
Yes, ma'am.
To share your slides. What happened? Yes.
Is it visible, madam?
Yes, we can see, but bring to the slide mode. Presentation mode. Yes.
Is it visible, madam?
It's visible. Please proceed. Yes.
Respected Chairman, Co-Chair, esteemed delegates, I am Jay Nanjundan. I am at Wellington. I am working on white rust in oil seed brassicas. utilizing the hotspot conditions here. The topic for the discussion is the identification and characterization of Indian mustard accessions with novel resistance against multiple isolates of Albua candida occurring at Wellington. As we all know, among the 7 oil-resistant brassicas cultivated in India, Brassica gentiana contributes more than 80% to the both area and production. So our main focus in germplasm screening breeding activity mainly on Brassica gentiana. So coming to the major diseases affecting Indian mustard, the sclerotinia stem rot number 1. Then other diseases, alternative blight, white rust. Now the powdery mildew also emerging in some of the dry tracts like Gujarat and non-traditional areas like Karnataka and Maharashtra. Coming to the white rust, this is when it occurs at the reproductive stage in the staggered formation, the yield losses are very high. So coming to the status in white rust resistant breeding in India, the entire Indian gene pool is susceptible to the white rust. So the Indian earlier breeding program, they used the 2 East European lines like Hira and Donskaja to evolve some varieties. Now we have identified 3 genes have been mapped. One based on Hira, another Donskaja. The third one is based on the Chinese line Tumida. Worldwide, there are 13 races of Albua candida have reported, of which in India the race 2 is mostly infecting the B. gentiana. Utilizing the East European lines, few varieties have been developed, like Pusa Mustard 31 from IRA and DMH 11 from the DU campus, but still not to be done because always there is a variation in the Albua candida new isolates. The profiling, the systematic profiling of which not done in India so far. So what approach we had is to bring the entire germplasm from the gene bank and screen at Wellington hotspot and to identify the multiple resistance against multiple isolates. So as we all know, the Wellington, it's a very natural hotspot for many brassica diseases. It comes naturally, even with a little bit irrigation management, we can bring nain score. So this condition, utilizing this condition, we are evaluating large number of accessions. So we brought over 7,000 accessions from the National Gene Bank, which includes indigenous collections, exotic collections, breeding lines, etc. First, we evaluated all these 7,000 at Bharatpur. Then we brought to the Wellington, the standard scoring method, the infector rows. We used at Wellington, we are continuously scoring during the both season because we can grow the crop at Wellington throughout the year. Then we repeated this evaluation for last over 10 years to identify the stable lines. After identifying the stable lines, we used certain markers already tagged with the resistance locus in Hira and Donskaja. So this is the program. We started with 7,000 accessions at Bharatpur. We narrowed down to 2,100 at Bharatpur. Then we brought 1,900 to Wellington, out of which we got only 86. So out of 7,000, we ended up in 86 resistance to Albua candida at Wellington. So this is what happened in Wellington. Up to Kharif 2019, we had 86 resistance. But after the monsoon rain of 2019, the new isolate arrived, due to which only 16 we could get as a resistance. Then we continuously screen. As of now, we have 35 lines with resistance to. We suspect that at this stage, with the preliminary findings, there are 3 isolates of Albua is operating at Wellington. So now we have 35 lines resistance to 3 isolates of Albua at Wellington, also for the against Bharatpur isolates. So in another work we published, we took 2,000 germplasm from the NBBGR. We evaluated at Ludhiana, Pantnagar and Hisar, out of which we could identify 168, 46 and 185 accessions resistance. Then across location only 27 were found resistant. Then under artificial condition at Delhi, using the Delhi isolates, we could identify 8. Out of 8, only 1 accession is resistant at Wellington for all the 3 suspected isolates. So in another attempt to augment the new germplasm sources, these were some leafy type of Brassica rugosa accessions explored from the Arunachal Pradesh, which represent the Indo-China border. So we could identify 12 out of 51 resistant. Then we also trying to collect some local germplasm. It is a typical one from the Ooty Hills, Nilgiri, which is having resistant to all the isolates continuously over the 5 years. So after identifying these differentials, we used the already reported 2 genes, that is Iran, Donskoza, the East European donors. We characterize the markers linked with them. In that evaluation, we identified 7 accessions. They were not amplifying these 2 already reported genes, but resistant at Wellington. So we suspect that these have some new locus other than earlier reported. This is a 430 base pair band we got for HERA based locus, another 750 base pair for the Donskoza based markers. So when we, among that accessions suspected to have the new genes, we made that crosses in F2 to test allelism. So we, at this stage, we can say that this W228 is non-allelic to the earlier genes based on F2 ratio. So this need to be mapped. Now, utilizing this resistant into resistant conventional breeding, we are attempting to augment the gene sources using conventional breeding. So 34 crosses we attempted. At this stage, we are at 51 single pair selections at F5 stage. So we are selecting for resistance, white rust, and also for the other agronomical expressions. So then last year we published in one paper in Plant Pathology Journal. For the first time, we identified powdery mildew resistant in Indian bee Gensia for the first time. So based using that genetics donor which has been registered with NPBR, we are making crosses to club both this white rust and powdery mildew. So we are at F1 stage. In the coming years, we'll be selecting for both the resistance in the segregated generation. So having identified these germplasms, now the ways forward, first we have to precisely screen these germplasm artificial conditions through single sporangium. Then only we can differentiate the 3 isolates occurring at Wellington. Then the racial profiling at Abu Candida in India, not systematically done, only isolated work happening, that need to be done. Then we want to molecularly characterize these accessions using the gene markers. Because recently our Delhi South Campus, they cloned these Donskoza based on tumidamase markers. So we want to, instead of flanking markers which we are using so far, we want to shift to the genic markers for more precise. Then allelism test to identify another locus, then we have to map, then ultimately marker-aided pyramiding of all the available genes. So this is about our presentation on white rust. Thank you. Thank you, madam. Thank you.
Thank you, Dr. Nanjandan. It was an excellent presentation, and it is a really, you highlighted in your summary that you really need to do race profiling. Because in many places people have attempted, but still the race picture is not clear for Albigo Candida. So I will appreciate if since Wellington is a hotspot area, so if that place, if you can really look into how the races can be identified, because people have done on different brassica species, and based on the pathogenicity on different species, they say it's a different race. Like maybe on Juntia, as you say, race 2 is there in your Wellington Center. So exhaustive research needs to be done, and you are collaborating with Dr. Lakshman Prasad also in our IRI, yes, some work. Yeah. Okay, now it's open for discussion. A quick question can be taken up.
Please.
Yes, Dr. Harbans.
Great talk. And I think that was the best material for the plant pathology courses to be used, because you can see hyperplasia and all sort of stuff. And but I think like technologies like Professor Kogel is developing, they could be useful in these kind of things, because they are xylem and phloem kind of related things. But they may be still away. But in the meantime, I think Professor Agarwal said that you have identified the hotspot, and just work as a nation to get the material and get it tested, and which you are doing it well, and maybe get more advice from seniors like Professor Agarwal. Great.
Thank you.
Thank you.
So can I introduce the next speaker?
Yes, sure. Welcome.
Sorry, ma'am. Ma'am, can I present at that time?
You, Neeraj? Yes. Okay, Neeraj, you are ready with your presentation?
Yes, ma'am. Yes,
with permission of Dr. Harbans, we will take him. Dr.
No
Neeraj
problems,
Dwivedi?
no problem. No
Yes,
problem.
yes. ma'am.
You kindly restrict to the timings allotted to you.
Sure.
10 minutes.
Okay, can I
Proceed.
make one point? Because
Yes,
we
sure,
have some
sure.
time saved. Just
Yes.
for like we are now going to go and retire. When you have given 10 minutes
Yes.
or 12 minutes, don't make your slides more than those minutes.
Right.
Even less than those will be better. And try to think through your slides. You're not telling your whole life history. You just are trying to if the message doesn't go, you have to make sure at least one message gets into the head of people who are not working in your area.
Yes.
So that's what I just, because there are 30 participants still, I wanted to make sure it's important.
They remain.
Yeah, please.
Yes, Neeraj, you,
Share your
Neeraj, you
slides.
just share your slide.
Yes.
What happened? He's not able
Sir,
to
my
share.
PPT is not showing.
You should have opened and minimized it.
Okay, I'm trying it.
You know?
Did
Okay.
you submit
I
here?
have, no, sir. I have opened it and I have minimized, but when I
Now
am sharing,
then you share and then it will come on the screen and double click that.
yes, ma'am. Yes, yes, sir. Yes, ma'am. Is it visible?
Yes, you
ma'am?
bring to the slideshow. Yes, please begin quickly. Yes.
Okay, very good evening to all of you. Honorable Chair Dr. Rashmi Agarwal and all the eminent speakers and participants. I am Neeraj Dwivedi, and today I am going to present my work on molecular breeding for anthracnose disease in Chile. And my topic is parental polymorphic survey and screening of after-mapping population derived from a cross between Butylacia and IV535 against anthracnose fruit rot disease in Chile. So this is a part of work which is going on under the project CRG project funded by CRB, and Dr. Lakshman Prasad, principal scientist in plant pathology division, is collaborating in this work with me. So, as you all know, that anthracnose disease is caused by a Clathrin-Mediated species, and it is a pre as well as post-harvest disease, which causes yield loss up to 50%. And we all know that India is the highest producer, consumer, and exporter of chili crop. And in India, Coleotrichum capsaicin, as well as Glosporites and Acutatum, they are the major causal organism of anthracnose disease in Chile. And till date, no resistant cultivar available. And so in 2014, this first time reported from IVR, Indian Institute of Vegetable Research, that Butylacia has resistance against anthracnose disease. So we have done some preliminary screening, and we also found that it has good resistance against anthracnose. And then when I was visiting the Northeast region for another project, so I saw there was a huge diversity morphologically in fruits as well as in plants. So I collected different Butylacia lines from different parts of Northeast. So you can see in this slide there is a huge diversity present in the fruits of Bujulakia. So then we have done screening of Bujulakia against Coletoricum capsaicin and gliosporides. So this is showing in vitro screening using fruit puncture method. So so in this, you can see that there is so much diversity in resistance. So some of Bujulakia lines showed very less resistance against the disease. So out of 24, we have collected and we have grown here, and after 2-3 generations, we have do the screening. So a few lines showed very high resistance against both the strains. And we have taken some high-yielding elite varieties as control or as susceptible varieties. So we have crossed Bujulakia. Accession number is CCA0164 with IB PBC 535. It is also called Kasi Shinduri. It is developed by Indian Institute of Vegetable Research, and it is having very less pungency, or you can say no pungency. So we have crossed and we have developed a F2 mapping population, and by using microsatellite marker, we have also tested the hybrid purity testing. Right now, these are the F2 individual lines. In greenhouse, we have to keep it in greenhouse because otherwise outside they could not survive because it belongs to northeast region, so the climate is quite different. We are doing genotyping using microsatellite as well as GPS genotyping by sequencing based marker, SNP markers. So here I am showing the results I have got from microsatellite screening. So I have used 380 reported microsatellite markers, which is distributed across the chili genome, and we have got 72 distinctly polymorphic markers, which is 18.94% were found among the parents. And all microsatellite markers amplified in the range of 150 base pair to 350 base pair. And actually, the F2 seeds have very low germination percentage because the outer seed coat is very hard, and we are doing interventions to raise the germination percentage. We have done the screening of few F2 lines. So 37 F2 individuals were screened against Coletoricum capsaicin and gliosporides, and 13 F2 lines were found symptomless. 6 showed very high resistance, and 2 F2 lines were found moderately resistant. 6 showed moderately susceptible, and 10 F2 individuals displayed high susceptible reaction. However, these are preliminary results, and more F2 lines are being generated for the mapping as well as phenotyping for anthracnose resistance. The polymorphic microsatellite markers and initial screening result will be utilized for the identification of QTLs against anthracnose resistance in chili. Thank you so much for your attention. Thank you.
Thank you, Dr. Neeraj.
Thank you.
For timely finishing. One quick question can be taken.
Unshare the screen,
Open.
please.
Yes, unshare.
Yep.
While unsharing, Neeraj, I can ask you one question.
Yes.
Good work. We just look at how does vitamin C goes along with these resistant sources in chili.
Yeah.
It's part of our life every day, and we can't live without chilies,
right.
Especially green ones. And I think the other thing is that it's. I think you're taking a good approach, but at the same time, look at a bit more laterally too, how you can improve it.
Yes.
Yeah. Okay.
Thank you. Now, Dr. Bariana, you can take up further.
Yeah. Because how are you?
Sir, how are you, sir?
Good, good, good. Because when I heard Dr. Nanjundan talk, is he based in Wellington as well?
Yeah, yes, sir, yes, sir.
Yeah, so I thought the first thing I will do after the COVID is I will come to Wellington.
It's long,
Yeah.
and Dr.
So,
and
but
Madam also.
look, let me introduce you. Because is one of the key persons at the Wellington station in the Rust program, and has spent some time with us in Australia. And I must say that he's one of my key links over there to do Indo-Australian Rust research,
Yes, sir.
and has been a great research partner. And we have published together, but at the same time, rather than going into too much of detail, I must say that because keep up with good work.
Thanks, sir.
And don't get carried away.
Sure,
and important thing is we need people to stay there and actually keep up with the sort of keep the work going. Otherwise, sometimes people leave and the work is not finished.
Thank you,
And
sir.
never, and then the whole thing never grows.
Thank you, sir.
Dr. Vikas, are you sharing from your side?
Yes, sir, I'm sharing, sir. Yeah.
Okay, thank you.
So all the best, and please go ahead.
Thank you, sir. Thank you, sir. So is it okay, sir? Am I audible and the screen has been shared, sir?
Yes, you are audible.
All good.
Thank you, sir. So, thank you, Dr. Bariana, for those good words. So respected chairmen, scientists, and other scholars. So I welcome to my presentation on marker-assisted pyramiding of leaf rust, stem rust, and polymorphism genes for durable resistance in wheat. So I am a little bit afraid because one of the my chairman, Dr. Bariana, is my teacher along with Dr. Urmil Bansal for my to do my visit to Australia, and I learned a lot of wheat and particularly rust sealing resistance from Dr. Bariana and Madam. So if I am wrong somewhere, please correct me. Thank you, sir. And wheat, as you know, wheat, that's what we eat. So it's a staple food of India, and India is the second largest producing country with an area of 30 million hectares and a production of 107 million tons. So it contributes around 25% of the country's food grain production, which is highly essential for food security. So that's the good part. And the sorry part or the bad part is that, so we have got a yield plateau for the past, you can say, for the past 1 decade, you can say the yield is plateauing, but at the same time the population is increasing. So strategy is to increase or sustain the biomass so that yield can be maintained. But the problem is it's not that easy. We have got abiotic and biotic stresses on the way. So the abiotic stress is mainly in the form of climate change, which I am not going to talk. And then the next one is the biotic stress. So there are a lot of biotic stress in wheat, out of which rust plays a major or a significant role. So depending upon the environment and the genotype, the rust can cause a yield loss of ranging from 1 to 100% because the range is very high. So it can cause anywhere between 1 to 100. That's the significance of wheat rust. So there are methods like easy way in the use of fungicides, but in the short term, it could be a solution. But in the long term, is it sustainable? That's the big question. So in India, you can consider that around in the stem rust is a major problem in the central and peninsular part of India, so which occupies around 7 million hectares. And at the same time, we have got leaf rust, which is present all over in the wheat growing areas of India. And now the currently the powdery mildew is gaining importance in the recent years due to the climate change and also due to some of the cultivation practices. So for this, how we can control? What is the approach you can go? It's a major approach is main approach is the host resistance or the genical approach, which is considered as the most reliable, economic, and environmentally safest and sustainable way to manage the disease. So it's nothing but prevention is better than cure approach. So the objective of this study is that marker-assisted pyramiding of the highly resistant stem, leaf, and powdery mildew resistant genes to make the host become resist to the diseases. So the materials and methods we have used is a combination of genes rather than a single gene. So as already the air speaker like Dr. S.C. Patel has told that it should not depend on a single gene. You should go rather for a combination of genes. So the use of resin parents, which is already been crossed, which is already having the SR36 gene, PM linked to the PM6 from Dr. CM Sivasamy, who is head of the station. So I would combine these SR36 PM6 with other stem and leaf resin gene. So we have used SR24, which is linked to LR24, which is still giving resistance, and SR26, which is now getting popular among the wheat genetics because it's highly resistant to UG99. So with this approach, we have gone for two selection. One is sealing reaction using sealing reaction sealing stage at IAWBR kernel, and Alpine reaction combining the leaf rust and stem rust at Wellington. So here we used to score using combining the response on severity using pustule type and also severity. So up to 20 is considered as resistant, and in seedling 0 to 2 is considered resistant. And probably we usually go for serine best scoring of scale 0 to 9, 0 to 5 is resistant, and 6 to 9 is susceptible. So for unit based selection, we use go for marker for 36 and 24 and 26. So the pyramiding, we usually taken the recipe parent, which is already having 36 PMCs with the dwarf kite, which is again Australian line, which consists of both SR24 and SR26. So the F1 is crossed with the respective recurrent parents. So we have taken a series of recurrent parents, 9 recurrent parents. So that will be respectively crossed simultaneously, and gone for 3 blackers BC3 F2. In F2, we have gone for a mass selection, genome-based selection, and also phenotype-based selection. So then we just selfed again to get a stable lines at BC3 F4. In BC3 F4 also we have gone for again phenotype-based selection and also mass selection. So in doing phenotype selection, we will go for rust resistance for leaf rust, stem rust, and powdery mildew, and also we will go for yield attenuating traits. So we won't select the lines which is poor yield, poor yielders. We usually go for high yield than the recurrent parent. So this is the result which shows the selection, the phenotype selection and sealing and the alpine station for stem rust and powdery mildew. You can see the recurrent parent and the HW numbers, which is the interghost line. You can see that it has got the recurrent parent, or the initial parent has got 3 plus susceptible, but the susceptible parent, the interghost line has got 1 plus. So it is showing resistant reaction. So there is similarly in the case of adult plant, you can see the susceptible parent is around 40s, but the susceptible parents, the interghost lines are around 3, you can say. So that shows that these genes are giving high resistance to the prevalent rices. So this is a series of crosses interghost lines. You can see are 9 interghost lines in different backgrounds. So most of the backgrounds are giving resistant reaction. Similar is the case for leaf rust in the sealing stage and also on the from the field stage. You can say the susceptible parents are giving the susceptible reaction of 3 plus in the sealing, and interghost lines are giving 0 flag or 1. And the same situation is there also there in alpine reaction. You can say it is around 40 to 60 years in alpine reaction, but it is free in of the interghost lines. So there are some of the pictures showing the resistant lines and susceptible parent. You can have clear difference between the susceptible and the resistant one for leaf rust, stem rust, and also for powdery mildew. So this is the molecular conformation for SR36. So SR36 is not expressed uniformly in all the backgrounds. Only in as a LOC1 and as a MACS 2496 we can have the full expression, and in other 2 varieties it is having only partial expression. But rest of the lines, it is not showing any expression. So there is some background effect, or it's not fully expressed in the other backgrounds except in LOC1 and MACS 2496. But when you come to SR24, it is expressed uniformly in all the backgrounds. Similar is the case with SR26 also. It is expressed similarly in all the backgrounds. So if you score for the presence and absence of the genes, you can see. So in HW5067 in LOC1 and HW5068 in MACS 2496, we have all the 3 genes expressed. But in rest of the backgrounds, you can have only SR26 and SR24. So SR30 is not expressed. So it may be due to any background effect or maybe some other reason. So Dr. Barena has to give some throw light upon it, because it is giving resistant reaction due to other genes, but it's not showing the presence of SR36. Maybe due to, I suppose, maybe due to some of the background effect which is not allowing the genes to express in the other backgrounds except in LOC1 and MACS 2496. So the take-home message is that the host resistance is the effective management strategy to control these rust diseases. And in this case, the population size matters. So we have to go a lot, since it's a 3 gene, combination of 3 gene, we have to grow a large number of genotypes or plant lines to select the desired combination. And the background of recipient gene is always a problem, so it won't be expressed, it won't give the same result for all the gene. So in this case, SR24 and 26 is expressed properly in all the backgrounds, but 36 is not having expressed. So in mass molecularization is the best method, independent of pathotype and environment. So there is no need that the environment should be present and pathotype should be present. We can select the genes based upon the molecular data. And these 2 lines will be either included in the acrylic trials, and also, or else it will be also in genetic stocks, so that other bidders can use this as a gene source for the transfer of these genes. So I thank IRA and my colleagues in IRA and also in IRA for performing this work. Thank you.
Please unshare slides. Unshare slides, please.
Yeah. Yes, madam, if you permit me.
Yes, sure.
1 minute or so, just for the audience as a sort of total plant pathology community, not only Rust. I think if we look at the collaborations, Professor Watson, who retired in 1986, I think, or maybe 2 years before, if I don't remember the date properly, he visited Wellington in 1970s, and all that, Dr. Brahma, Dr. who was the other person, main one? Just tell me, I'm forgetting the names.
Dr. Menon.
Dr. Menon. And Dr. Menon, when I visited, he actually told me those stories. And international collaborations are very good to build up all those things. And I think Wellington, Dr. Tomer, and all those things have been built very well to deliver to the wheat community. And just one thing to add from the wheat community, SR26 translocation has 2 genes, SR26 and SR61. So that's why it was not defeated so far. And people are trying to associate and reassociate. So, but you can say that already nature has given us something. And what I want you to do, because is have large number of F2s, try to get better yield out of it.
Yeah, just this water
So
line.
if you can give better yielding line out of the SR26, I can send you even small. You have the small one. Don't use the old one. Small one, you already have it. We will talk later. But I think even small one has 2 genes.
Oh.
So just try to get the yield. And for the others, I would say that together, if we look at the host and pathogen sort of things together and work along with other colleagues, the main thing is never look at like this. Look at like that. World is there to embrace, not to like this. So if we work together, we can achieve a lot. If we are actually doing it individually, we can not progress. That's what. Although these days IP laws are making it difficult, but still we have to bend them over.
Sure.
Thanks, madam.
Thank you, Vikas. Thank you, Dr. Bariana. Should we move to the next speaker?
Please.
Monica, Kalyan, I was not finding her.
We can next ask Mr.
Next, Asha Jyoti.
Yes,
Asha Jyoti Mishineni.
Ma'am, I have sent already voice recorded PPT. Can you share?
We are not able to, actually, we are not able to play that, I suppose.
No, Asha Jyoti, did you submit?
Yes, sir. Or otherwise, I will share from here.
Then I'll try from here, okay?
Or else I will do, sir.
I think it's better if she shares. That will be better.
Okay.
Yeah. Because she's there. Yes,
okay, you share.
Asha Jyoti, you move.
Yeah. Okay. Good evening, respected
Make it
dignitaries.
slide mode.
I'll just keep, sir. I mean, from second slide, I will just play, and for the introduction, I'll just myself narrate. I'm here. This is Asha Jyoti, and I'm here to present my work on black pepper endophytic Pseudomonas cutida, BP25, which modulates defense elicitation in in rice against fungal blast disease. Since I have kept the voice recorded one only, I will just.
Voice recorded you are playing?
Yes, ma'am.
We can't hear. Sound is not coming. So better you present now.
Okay, ma'am. Then
you present.
Okay. But without voice, I'm not having. That's why I just kept like this. Oh my word.
Otherwise, what you can do, you can just go to his slides, and I think there is a switch coming that the volume can be reduced further, so it can be muted here. And this bar.
Can you play your slides from your computer or something?
You may be having unrecorded version of your PPT, Asha.
That she doesn't have. I think she has to create. So only one command is required, that go to the slideshow and then just record all the.
Yeah. My God.
That's great.
Everybody got this confusion, no? The played and the.
I think the message for next time is that never record it.
Yes.
Never record it.
Never record, because sometimes compatibility patterns and.
Because then people.
Yes, Asha Jyoti, you are muted. You unmute yourself. Yes, now. Proceed.
Ma'am,
Asha Jyoti, you can present.
Still, I could not make it. Otherwise, I will stop sharing. I will recorrect it and then I will come back, ma'am. Please go on.
You are the last speaker left. So all the people may leave.
Just try from That's here. Okay.
You speak like that. We have.
Just tell us the story.
Without,
Tell us the story.
Without slideshow, yes, ma'am. Without slideshow, I will just share my presentation.
Sure.
Okay, like this only I will keep ma'am.
Okay, no problem.
Thank you so much.
Yeah.
My study is based on a novel endophytic bacterial strain, that is Pseudomonas cutida BP25, which was originally isolated from black pepper cultivar Panur 5 as a root endophyte from Kerala in the way back 2009. Now, in previous studies, this isolate has known to produce some biocidal volatile compounds against major plant pathogenic species, and also it was known to colonize taxonomically diverse dicot model plant, that is Arabidopsis thaliana, and also it has induced phenotype alterations in the roots of Arabidopsis, and it was known to trigger salicylic acid mediated defense pathway by altered gene expression. Based on that previous experience, we thought like we hypothesized that this bacteria can colonize taxonomically diverse plant species. For this study, we have taken the strain and we have reconfirmed the identity through by growing it on LBA and on antibiotics amended media, that is rifamycin and gentamicin, and this isolate is also GFP tagged through TEM analysis also, and by sequencing 16S rRNA and Pseudomonas cutida specific markers, that is TPIA and TRPC markers, we have confirmed the identity of this particular bacteria. And later, we have tested the endophytic behavior of Pseudomonas foetida in rice. Just a minute. For this, we have soaked the rice cultivar Pusa Basmati 1 seeds in decimal dilutions of bacterial suspension for 24 hours, and the same seed was grown in sterile petri plates under greenhouse conditions for 15 days. After 15 days, the plant phenotype in terms of root length and shoot length was measured, and interestingly, we found that a higher concentration, the bacteria, the endophytic bacteria, able to trigger root phenotype alteration in rice also. And however, there was no significant effect on seed germination. And later, through plate count assay, no, I could not show. Through plate count assay, we have estimated the internal population, and we found nearly like 3 to 4 log CFU per gram tissue of the internal population of endophytic bacteria in rice seedlings, which were treated with the same bacteria. And also through PCR based detection assay, we found where we have targeted the GFT gene, and we that yielded 142 base pair GFT amplicon, and we further confirmed that this can colonize taxonomically diverse crop species like rice also. And through again it is later through quantitative PCR assay, we have found after 7 days the internal population was like in correlation.
If it's freezing, can you actually please, don't worry about the slides, just say 4 or 5 points which you think you want to tell us.
Okay, sir.
Yeah,
okay, sir, I'll
because this is your work, and you don't have to worry about this technology to work. You can talk normally.
Okay, sir.
That will be good, because then we can actually catch it better rather than playing with technology.
Yes, sir.
Yeah.
In a nutshell, my study is that black pepper associated Pseudomonas foetida, upon seed priming, it will induce root phenotype alteration in rice cultivar Pusa Basmati 1. And also it was known to colonize internally the rice, and it was confirmed through PCR and confocal laser scanning microscopy and RT-PCR study. In RT-PCR based absolute quantification, we found after 7 days treatment with the Pseudomonas foetida, the internal population will increase with the increasing concentration, external concentration of bacteria treated to the seedlings. But after 14 days, the internal population will be regulated or stabilized, unlike any pathogens. This means here we can see the clear-cut difference between pathogen and endophyte. An endophyte can, internal population, it can regulate, and the pathogens which cannot regulate that cause the damage to the plants. And secondly, our second hypothesis is that the altered root phenotype, or the phenotypic expression or shortened root phenotype, that is may positively influence the rice in defense regulation. Here we have taken the blast disease pathogen, that is Magnaporthe oryza. We already know that it produces biocidal volatiles. So through volatile and non-volatile assays, we found that it can inhibit the mycelium of rice blast fungus. In volatile assay, it was nearly 70% inhibition we have found, and here in case of non-volatile assay and direct antibiosis, it has produced nearly 40% inhibition. And through in vitro assay, that is through seedling greenhouse experiment, here we have found nearly like 70% less, 40% less inhibition over the control plants. This is an alternative management strategy for rice blast management through this endophytic bacteria. This is in a nutshell, sir. And the future forward for this study is whether in altered defense gene expression study, we found that PR 1.1 gene upregulation we have found, and PR3 downregulation also we have found, and the ethylene responsive genes which are involved in development of rice, that is ACS6 and ACO4, also downregulation we have found. And that defense altered defense expression gene study has given a clear-cut like explanation of this endophyte mediated defense elicitation, and also as well as root phenotype alteration study. And also in case of biochemical enzyme based assay, we found like peroxidase and total phenol content was upregulated in endophyte treated seedlings of rice. This is my study, and I acknowledge my entire team for their sincere efforts, and thank you for your patient listening and giving me this opportunity. If you have any queries, I'll happy to address.
Please on share screen.
I'm so sorry. I'm so sorry, sir. I did a mess.
That's fine, madam. Can I say something?
Yes, sure.
I think if you're talking about rice blast, and look at wheat blast as well.
Sure, sir.
That's killing the industry at the moment.
Yes, sir.
And please, madam.
Yes, Asha Jyoti. I know this work. I think you carried out in the division.
Yes, ma'am.
Good piece of work. Just one clarification. At that time also, I think this question was a reason that Pseudomonas putida sometimes is pathogen also.
That is Pseudomonas aeruginosa.
Aeruginosa, not putida?
Not putida.
Okay, it's an absolute endophyte?
Yes, ma'am.
The seed priming gives you like protection from blast.
Yes, ma'am. We have to give booster dose also.
Booster dose also. Okay.
Yes, ma'am. We have to give.
So it's a good application part, and it really works. It's a good technology which can be proven and then taken further.
We are taking it further through transcriptome study also.
Yeah. Asha Jyoti is a very nice presentation. But sorry, you could not share the slides. We could not see your...
that voice recorded thing.
Okay, any other query or clarification or question? Professor Kogal, Professor... Oh, I see Professor Niranjana. Welcome, sir.
Just a quick clarification, Dr. Agrawal, to our speaker, Aisha. The Putida certain strains can be opportunistic human pathogens. So Dr. Agrawal is correct. So not to say that your particular strain is going to be a human pathogen, but that would be flagged if you are going for commercialization and things like that. So just check with the information that's available. It's a opportunistic human pathogen, but that's a very broad statement because there are many strains of Putida.
Many such.
Yeah.
So definitely one can look into the toxicological data can be generated, and then one can go for commercialization. Okay, fine. I think we have presentations. If Kolam or Monica Kolam is there, we can take her. Otherwise, we are at the end of this session. Professor Niranjana, anything to say from your side?
You are on mute.
No, he is unmuted.
You have conducted very nice session, and they have given all presentations very well.
Thank you. We must appreciate Dr. Bariana. You will join with me in really appreciating all the presenters.
Yeah.
And definitely our keynote speakers were excellent.
But
excellent. Everybody. Invited speakers also had a very novel ideas, concepts, and all oral presenters. Very good.
Professor Shekhar Shetty lecture has been done.
Dr. Shekhar we called upon, but he was not there.
He has not turned up, eh?
He has not turned up.
Okay.
We looked for him and we announced also, but we could not hear.
So, ma'am, then I think.
Yes, we can conclude, Dr. Kalyan.
May I request both the chairs to put their remark if anything is left. I think we had a very good session, nice session. I am enjoying because I am rice worker, but still there are a lot of inputs and in-depth understanding we share. And I think that is a good thing we are learning. So if you have any remarks, particularly specific to the any kind of overall session, so just put one by one. Cheers.
From my side, I've already congratulated all the speakers, and they were excellent presentations on diverse crops, diverse pathogens, and many different areas from the conventional resistance, race profiling, and also looking into very deeper management strategies like double-stranded RNAs, and also what Dr. Fernando presented. So from my side, I have concluded and said that this session was really very nice, very fruitful, and also on the emerging disease of Fusarium head blight, which Dr. Saran talked about. So although it is there in temperate countries, we hear from European and other Canadian and USA, but now this is another emerging pathogen of wheat. So we must work extensively on Fusarium head blight. And I'm very happy that Dr. Harbans Bariana extended his help and germplasm sharing. So that's wonderful, and one can really have a networking program or anything with Dr. Fernando and Dr. Harbans Bariana. So thank you so much, Dr. Harbans and Dr. Fernando and Dr. Kogal, for really a very nice session, and all the invited speakers, Dr. Robin, Dr. Saharan, then Dr. Divedi and Nanjundan, and a very emerging again problem, and Dr. Vikas and Asha Jyoti. So it was all very diverse and very nice presentation, everyone. So thank you for giving me this opportunity. Dr. Harbans, you have any?
Harbans,
you can conclude and say.
Madam, you have said all, but I think it was a really great pleasure to be chairing this session, co-chairing this session with you. And I think we had really great science presented in this session. And I would say that starting from lab to the land, it was a really great theme, I would say, in this sort of session. And crops covered, you know, which we eat every day, chilies, and other sort of methodologies which are used in different things. I think I was really impressed with all the things, and you can imagine that sometimes I try to work on turning my mode off, but I could not. I actually put full attention to every person's presentation as a co-chair, and thank you very much, so much to learn and to absorb, and let's be in touch and talk to each other. Don't worry, chili person can talk to a wheat guy. And likewise, at the end, we have to counterfeit our enemies. And we can devise strategies like Professor Kogel's dsRNA can kill everything, you know, if we succeed in that properly, you know, that can be applicable, as he has shown, to so many pathogens. And Professor Fernando's things, you know, how he has shown how the things can develop, and Suvash presented, which is, you know, all the Indian system, and all the brilliant fellows like Saharan and Dr. Saharan and all the younger generation. I'm quite proud of it. Being an Indian-born Australian, I wish you all the best.
Thank you. Thank you, Dr. Bariana. And I think on behalf of organizing committee, as well as on behalf of Indian Phytopathology Society, we are really thankful to have both of you as a chair, and both of you are known for your recognitions in the area of wheat diseases. And I think we went very smooth, and though there are some technical snacks in the sessions, but somehow we could manage it by speakers. We are really thankful to both the chairs for your kind consent to us, and kindly chaired these sessions. And We are also thankful to particularly the keynote speakers, Professor Kojal, Professor Fernando, and Professor Bharadwaj. So you really made excellent presentations, and based your expertise field actually gave us a lot of input in understanding the basic science in plant pathology. And I hope that most of the participants had enjoyed your presentations, keynote address, and I think that is the ultimate benefit of these online seminars. So we are also thankful to all the invited speakers. I should name Dr. Rubin Gogoi, Dr. Saharan, and actually Dr. Sethi could not make it. So also thankful to all speakers, particularly Divedi from Terry, then Nanjundin from Wellington, Vikas from Wellington, and Asajoti, she is our student, previous student. So it's really a very interesting session we had. So with this, I again extend my gratitude and thanks to all of you for kindly making this session a very fruitful and enjoyable. Thank you very much.
Thank you, Dr. Kalyan. Thank you, Dr. Harbans. Good night, Dr. Harbans.
Thank
Dr.
you very
Fernando,
much.
Dr.
Thank
Kogel.
you.
Everybody, hey, good morning,
Nice
good night,
interacting
good
with all
afternoon,
of you.
wherever we all are.
Yeah,
Dr.
yeah, we are
Fernando
one.
may be having more.
We are all. That's wonderful. Dr. Niranjana.
I should not forget to thank the participant. I think I should specially mention Dr. Madhuvan Gopal.
Madhuvan Gopal, yes, special thanks.
Niranjana. I think this is a really great inspiration for us, that those who are not, you can say, participating,
Outside,
but
but still.
and listening important talks. So this is really, if I miss anybody, just excuse me. And it's really a great thing that you participated and enjoyed the session. Thank you very much.
Thank you, Dr. Kalyan, very nicely. Thank you.
Thank you, madam.
Yes, thank you. Welcome.
Thank you, Saran.
Oh, Dr. Jayalakshmi. Anguru. Okay. Thank you, Dr. Niranjana. All the best.
Dr. Suresh.
LM Suresh, I think I saw somewhere message. You know?
Yes.
Thank you, Dr. Suresh.
Okay.
I could see you, Dr. Pun. Many people had joined.
Yes.
Thank you, each one of you. Nicely. We enjoyed this session.
Okay, ma'am.
Okay,
Thank you.
Kalyan, we will leave now.
Thank you.
Thank you.